long time. Okay, I think we're going to just get underway again. So for people who are getting organized.
Well, I hope everyone had a nice lunch and is fortified to go into our afternoon. We figured we'd do something similar to this morning where we'd have the presenters give their talks, and if you had a question that was directly pertinent, to ask it then, or otherwise we'll postpone toward the end and then have a group discussion. Our first presentation is by Gary Andersen of the Lawrence Berkeley National Lab and University of California, Berkeley, and he is going to speak on the built environment microbiome.
Thanks, Joan.
Go, Gary.
Okay, I will go. Thanks, and thank you for inviting me here. What I want to talk about today, hopefully, can you hear me fine? I want to talk about kind of a historical perspective from a science, from a microbial ecologist perspective. So what was the reasoning behind why we were interested in looking at the air, especially the indoor microbiome? So the outline then of what I'll be talking about is first setting the stage, work that was done initially on looking at microbes in the atmosphere, what factors influence dispersal, and from there, what are the sources of the microbes that were in the built environment? How do they differ from the outdoor air? And then doing some, Paula mentioned some of these, and I'll go over some of these and some additional early, very seminal studies that talked about how the microbes, what were the sources of the microbes in the homes and in other environments like hospitals. And if I have time, I'll try to keep on schedule here. I'll talk very briefly about how we look for other factors such as viability of microbes. So to start off with, I want to talk briefly about the outdoor microbes, because I think to understand what's going on indoors, you need to understand also what's going on outdoors. And so the atmospheric environment, the aero environment, is very unique from other environments. It's not like soil, it's not like water, it's not like plant leaf surfaces. Microbes, for the most part, aren't born, live, and die there. They spend a portion of their lives there. Some organisms either by accident, others on purpose, but organisms by and large are there just for, at the most, portion of their lives or a few life cycles. Not there for hundreds or thousands of years continuously. So population in any one place in the atmosphere it's a sum of a couple of different factors. The immigration, what's dispersed into the atmosphere, the emigration, what's then removed from the atmosphere, and it's also within here what grows, what dies. And so I mentioned some of the sources of microbes already. How are these dispersed in? Many times it's just accidentally entrained by wind. Other times there's active dispersal mechanisms because some organisms actually have a portion of their life it's very important to be in the atmosphere. And then removal from the atmosphere, even if you're in there, what goes up must come down. So even if you want to stay in the atmosphere, often you don't have a choice. You just come back down. So here's a kind of a stylized picture of Earth, of the air. And so many different sources of microbes. Microbes can come from the sea, from plants, from cities, from different types of plant surfaces. But the atmosphere is a very unique and it's a harsh environment. It's an extreme environment. And so some of the factors which make this such an extreme environment are UV radiation, a very important factor in survival of organisms. And other factors, including just that in air, even though there's precipitation, for the most part, there's not a lot of water in the air. So just the desiccation stress is also very important. And we heard from David this morning about some of NASA's programs. And since I wasn't going to talk about this before, but organisms can be anywhere within the atmosphere, but NASA has put organisms into space, not only just in space stations. They've actually purposefully, one of his researchers, Kasturi Venkatasawaran, started out almost 10 years ago on the International Space Station. He put Bacillus pumilus out on the surface of spacecraft in a special experiment and had spores of the Bacillus pumilus out in space for up to 18 months. So these were put out in space in different forms, either shaded from UV or not. And when they came back to Earth 18 months later, the organisms which were shaded from the ultraviolet light, a large percentage of these spores actually he could grow up again. What was interesting is even with those that were subjected to UV radiation, there were many millions of spores, he was able to get about 18 spores to actually germinate after 18 months in space. And I think you would know more than me. I think this was a magic number because that's how long it takes to go to Mars. So I think that's why that time was chosen. Too much on that. When we're starting to look at, there's been a long time of looking at microbes in the air, but starting about 15 or so years ago, 10, 15 years ago, molecular techniques came in. So instead of culture-based methods, we could start to use DNA-based methods to look for microbes. And this was just an early study that we did in my lab, looking at microbial transect across California from east to west, from the coast to the mountains, in both a rural and an urban setting. And we just found a very large diversity of microbes overall. That's what these different bars are here. But we found some interesting things. We found that the urban environment had a much higher diversity of organisms, in this case, than the rural environment, and that as you went from west to east, you also increase the diversity. A little time after that, we had the anthrax outbreaks. And so Homeland Security ramped up, and one of their programs was looking at organisms in air. And so don't want to get into the politics of BioWatch, whether it was good or bad, because I would have opinions on that. But one of the things they had was a very large program, which they had these APDS, autonomous pathogen detection systems. And I highlighted a couple in Washington, D.C., mostly outside, sometimes inside in subways, things like that. And this was a very large program, $100 million per year, looking at basically what they looked at was 5 potential bioterrorist organisms, and that's all they looked at. But they had many false positives were detected each year. They really weren't looking at the background. It seemed like to provide, they were doing all this sampling. Let's look at what some of these background organisms are. So in my lab, we looked at some of these. In one study, we looked at 2 very close cities, 100 kilometers apart from each other, are San Antonio and Austin. Very similar ecological conditions, similar size, about 1 million people, similar environment on the outside. And what we wanted to do was we did 17 sequential weekly samplings for these 2 cities just to look at the source of variability. What was the diversity by time and location? And really just summarizing very briefly in this multivariate regression tree analysis, what we found was that the main driver of microbial community structure was time, the week in which it was sampled. And these are listed in order of important environmental factors. After week, the temperature, time of sampling, PM 2.5 content, and mean sea level pressure. So these were the important environmental factors. We saw it was mostly meteorological and time factors that shaped the outdoor environment. What was striking from here was the lack of the space. The city to city was really not that significant. These 2 cities were similar from 100 kilometers from each other region. It was more these atmospheric drivers, these environmental drivers that shaped the community. We looked at some specific organisms too, and so this was using 16S-based technology. And so we're looking at different organisms, and this is a 16S sequence of a mycobacterium similar to Mycobacterium tuberculosis. What's really important to remember in these studies is that when you're at a 16S level, you're really not looking at pathogens. You're looking at organisms which could contain pathogens or environmentally closely related species. And what we saw was that as we increased the temperature of the sample, we increased the amount of this mycobacterium that we could detect in our testing. So that was just to give you, a kind of a broad overview of what was going on outdoors. So at the same time that this was going on, that Paula was mentioning this morning, starting up this program, the human microbiome was coming up and showing some very interesting results on not just on diseased people, but what healthy people, the diversity of organisms. At the same time, the studies that had been done indoors were mostly on sick buildings, looking at disease, looking at moist buildings, not at healthy buildings. And so I think that was one of Sloan's really interests in starting a new area. The area of looking at sick buildings has already been taken care of, but what was the microbial ecology of what's going on indoors? And so what is some of the first seminal questions were, what are the origin of the organisms which we find in we spend 90% of our time indoors. And the hypothesis is that these organisms would come from the outdoors, or they could come from shedding from human and pet microbiomes. They could come from people into these environments. And then once indoors, is there selection from building specific factors? And so back to the idea then of indoor microbes. Are there microbes which are unique to the indoors? That is, microbes which don't occur on the outside, but which have adapted over years through selection and genetic drift to live just specifically in specific environment indoors. These were some of the initial questions that we're interested in on. And so I talked to you a bit about the outdoor environment. People are also a source. I mean, here's a good example of people coughing in a room or whatever, just spreading organisms. But, it's not just coughing. So work by Bill Nazaroff and other, Paula was talking about particles from people. And so looking at thermal emission, this is a thermal emission just coming from a sitting down person, and we are microbial soup, and there's organisms that are just constantly coming off of us, thousands, millions of organisms within a specific time period. As you go into a classroom, as you go into a meeting room here, we are just releasing millions of organisms. And I'll talk about some of the work Jack Gilbert has been doing on this. So one of the earlier studies that was mentioned in looking at indoor air was a study by the Berkeley group, the Bimert group, and this was by Tom Bruns. And he used a sequencing technique which was popular at the time, pyrosequencing, looking at shorter regions of sequence of intergenic transcribed space, or ITS regions of the ribosome. So this is looking at specific regions to identify taxa of fungi. And he could classify those taxa based on similarity to other taxa, either to 99 to 97, 95. And typically what's used is around a 97% identity from one ITS sequence to another to identify taxa. And one of the things they found was that as you went from the equator to temperate zones, the temperate regions had a much higher level of diversity than the tropics, which was counterintuitive. So this was a study of 72 buildings from 6 countries. And what he also found, what was interesting, is the function of the building in this study had no influence on the indoor air composition more than location, the outdoor. The outdoor environment had the most influence on these fungi. And these fungi were somewhat range limited. Those which were in the tropics were mostly limited to there, and so on. And this work by the Berkeley group was carried on. They did a much more detailed study in a graduate student housing area, an area of about a half kilometer by half kilometer, looking at specific houses. And as the title said here, what they found, they agreed that with earlier studies showing that the indoor air, the indoor fungi are dominated by outdoor air. And so what they did for sampling, this, they developed this new sampling technique to sample the settled dust. And this is incorporating a month's worth of dust on a sterile petri dish, and then looking at using pyrosequencing of the ITS region. And they found that, for instance, winter had a higher diversity of organisms than summer. They also found that even within this half-kilometer distance, there was separation by space, so that as you diverged from one location to another, even within this half-kilometer, you increased your diversity. So also want to talk very briefly on Jessica Green did a study on hospital microbiomes, what Paula was talking about on looking at on ventilation. And this is just from her work from showing that as you have more mechanical closed windows, mechanical ventilation, you had a higher level of pathogens than the outdoor air with the open windows somewhat in the middle. And this also you could see on community ordination, looking at the structure of the community from mechanically from closed window rooms resembled more people, whereas outdoor air resembled more outside environment, the dirt, and the open windows were somewhere in between. So this agreed, I won't go over all of this because Paula talked about this, but earlier reports had suggested that if you open a window in a hospital room, you had a healthier outcome. And this suggested a reason why. You increase your diversity of organisms indoor. Also, Jack Gilbert did a very interesting study. Paula talked a bit about this too, on the home microbiome, looking at 7 different homes on a very detailed level over a 6-week period on a daily level, looking at different parts of each individual occupant, plus many different locations within the home. And as she said, each family had a very unique fingerprint, and it would even change from if you went to a hotel room, you would overwhelm the signal of that hotel room within 1 day. A family would have its very characteristic signal. And this was done by a newer technique using a more high throughput DNA sequencing technique, which used even a shorter sequence, but it was you could get just millions and millions of reads. And so what he did here was he used a Bayesian type statistical analysis to show that that different locations like kitchen light switches, doorknobs, bathroom doors resembled more your hands, whereas the floor and of different places resembled more your feet. And each home was very distinctive to each other. He also could see from this study, this paper which published in Science in 2014, that when in one particular house there was a couple who had a boarder, and the couple had a much closer community to each other than their renter did. So showing just the very different unique interactions of how humans interact with the environment. Now, one thing you may be asking then is, well, some studies are showing outdoor air is important. Some studies are showing that indoor occupants the people are very important. And to an extent, it depends on the type of methodologies you use, many different methodologies you use, and also what you're looking at. So maybe fungi are different than bacteria. We're emitting many more bacteria than we are fungi, for instance. And so these are some of the questions that will be really important to further address in the future. Last study I want to talk about is some of the work that Jill Banfield did on necrotizing enterocolitis. And so this was a very interesting study, again, a very unique environment, a very precise, very sterile almost environment that's very clean, so very extreme. And within this environment, they were able to access different samples. And what she kind of was able to do here was to take an approach where she, instead of looking at these 16S sequences for either bacteria or fungi, she looked at everything. Because these were much simpler communities, she was able to actually look at the entire microbe, not just one biomarker molecule from the organism. So she could actually see strain level differences because she's looking at the entire genomic sequence. And so the hypothesis she had was that there was a potential outbreak of this necrotizing enterocolitis. Maybe there was an organism that was shared among these premature babies. And what she found instead was that almost all of the co-hospitalized, so these were necrotized NEC cases that were happening at the same time, had distinctive different organisms. It wasn't being caused by one organism. So there was no organisms that was shared from organism or from individual to individual. And you would not see this by a 16S type of, or any type of ribosomal-based survey. So it shows the value of some of the later techniques which are coming out. And this is kind of a complicated slide, but what I wanted to highlight, where this is from one individual over a time course of having the necrotizing enterocolitis and then going on antibiotic therapy. And basically, these are just the level of different organisms. But instead of having thousands or many thousands of different species, it's on the order of a dozen or so that the infant had at any one time. And these organisms are, for the most part, hospital-acquired pathogens. They're cleaning the surfaces all over in these NICU units. And these seem to be the organisms which survive that intense cleaning. And so these are the organisms which are colonizing these premature infants. So again, just kind of summarizing what they found that the communities were highly dominated by just a few organisms, but it was these hospital-acquired pathogens which seemed to be the first thing that would colonize and take over the microbiome of these infants. I just want to finish up, yes, I have time, very quickly on live dead then. So again, there's a number of different methods for looking at this, but this is a question that's been vexing in this field for a while. What if you have organisms, you're using these new genetic methods, you're doing different types of new sequencing technologies. New sequencing technologies are always coming out and finding more and more interesting things. But are the organisms that they're looking at actually alive? That's a key point too. And so some of the ways you can do this are using vital stains, live dead stains. And this is one case, PMA, Propidium Monoazide, for live dead discrimination. And so how PMA works is it's a very large molecule. It doesn't enter living viable cells. It can't cross the cell membrane. But on damaged cells, it can enter. It intercalates within the DNA. You can, with light, cross-link it, so it makes the DNA impossible to have amplification. So you can't amplify, and you therefore discriminate. And so we helped in a study by Jet Propulsion Laboratory. Again, this was with Parag Vishpamayan and Kasturi Venkataswaran. And so what they found here at JPL was that the PMA worked very well on these bacillus spores to discriminate live versus dead. They would mix a certain percentage of live versus dead spores, and what they would always see by analysis was just the live component in this method. So that was one promising avenue. And so that's kind of the summary of what I have to say, just kind of giving an overview of some of the questions that are coming up and some of the issues that hopefully you'll see in some of the next talks. But again, some of the questions that we are looking at are based on the methods that we used. And that's going to be very important, in future work, because we don't want to keep on reinventing the wheel. We want to make sure that even if we find new great technologies in the future, of course, that our current work is still relevant. So that's all I have on this. So thanks. Any questions specifically on this? Okay. Thanks.
Thank you.
In the human space, we're realizing that we cannot reproduce what's going on in lab to lab because the method, like there are even contaminants in some of the reagents we work with. Is the built environment community aware of these issues? And
Absolutely.
and they're trying to standardize that?
That's a very good question, and I think that's a very good topic. Paul has brought this up at many times, and I think that's a very important area to look at because, especially as you get to these very fine methods where you're just using just a swab, a little swab, and you're doing a whole DNA sequence based on it, and you're doing amplification methods. Sometimes, are you amplifying just some contaminant in one of your reagents, for instance? And that's a huge issue. And you know, it's been that way from the beginning. That's something I think would be very important to look at in the future. Thanks.
Thank you. Well, there'll be more time for discussion when we get to the end.